Excellent. My name is Lenny Teytelman. I'm one of the co-founders of Protocols.io, and it's my pleasure to host the intro webinar. So before we get started, just a few housekeeping things. First of all, as I just mentioned, are recording this webinar. We will send a link to the slides that you'll see and a link to the video with this recording to everyone who registered. So if there are any URLs, links, or things from the slides that you'd like, you don't need to try to take pictures with your phone or write down very quickly in notes the URLs. You'll have the slides and all of the links and information in them. There is closed captioning in Zoom that is enabled. So if you need to, you should see at the bottom of the Zoom control panel, you should see the button for turning on closed captioning, which is automatic transcription. At the end of the webinar, we'll have a Q&A. We keep the webinars attendees on mute, but please put your questions into the chat or into the Q&A, and I will make sure to get to them. And we do offer different webinars throughout every month. This is a webinar for those who are new and recently created an account on Protocols.io. So it's a general introductory webinar. We also offer an advanced one. We offer one specifically around academic use and getting credit and recognition. But whether you're from a biotech or pharmaceutical or from academia, if you don't want to wait for one of the webinars, this is an important link. We have a great help center, but also you can go to protocols.io/help demo and you will be able to request a one-on-one meeting with one of the scientists or customer support professionals on our team. And then you can connect for 15, 20 minutes or up to 30 minutes with a particular question or more advanced features. So it's not just these webinars. We have other resources, and the one-on-one demos are particularly helpful. I just clicked on the demo accidentally. I mean to still be in here. All right, so with that, let's get started. This is the agenda for today. I will introduce Protocols.io, why we built it, when we built it very quickly, and then we'll talk about sharing methods and the general problem that Protocols.io tries to address. We'll talk about where publishing is moving for those who are academic on this webinar, but the platform is equally useful for academic and industry, so it doesn't matter where you are at this moment of your career. It is something that is widely used both in academia and industrial settings. And then I'll do a quick demo of about 15 minutes of the main features of Protocols.io, how to get started with the platform, and then we'll go to the Q&A. Great. So, as I said, I'm one of the co-founders of Protocols.io, and my co-founders and I started the idea in 2012. We launched Protocols.io. It went live back in 2014. And there are two sides to Protocols.io. I'll show it in a second, but there is a public side, which is free to use, free to read, free to publish, and it's all open access. So there's the public repository, and there are over 23,000 shared public protocols on that side. And then there is the private part of Protocols.io. That's the part we charge for, is internal collaboration, internal use of the platform. And there's about 200,000 registered users and about 1 million people every year come to read the public protocols, and these are the 200,000 users that are creating the content and using the private side. As you can see here, there are a lot of worldwide, there are a lot of universities, and then there are government and biotech and pharmaceutical companies that also use protocols IO. Right? And our mission is to make it easy to share method details as you're developing um, the and doing the work as you're developing methods and doing the research and making it easier to uh, publish and making it easy to correct the protocols long after you've published them. So actually, the reason I co-founded Protocols IO is that after my PhD, I'm a scientist, geneticist by training, and computational biologist and hands-on experimental biologist as well. But it was during my postdoc at MIT that I spent a year and a half correcting just one step of the single cell microscopy method that I was working with. And at the end of it, I realized it's not a new paper, it's not a new technique, it's a correction of something previously published. So I don't get any credit for it. And everyone else who's using this technique is getting misleading results or has to spend a year or two rediscovering that single step change. And I became obsessed with creating a central place that makes it easy to collaborate and to keep people up to speed as you change. So even if you've published a protocol and it's part of a paper or not, long after publication, Protocols IO is a place where you can come in and share corrections and optimizations with everyone who's using that method. So I'll show that in the live demo. But as part of the introduction, what we said is there are two sides. There's the open access repository that's free to read and free to publish. And then there is the private side of Protocols IO. That is our business model. That is the part that we charge for. Um, a lot of universities around the world and companies have, if you're in a company that has subscription, you'll know about it. If you're in a university and you're not sure, you can navigate to the page protocols.io/for institutions. It's also available through the footer. And there's a drop down there that lets you check if your uh, organization actually has a subscription, premium subscription that makes this private use unlimited as well, right? But in terms of the classic problems with research papers and reproducibility and why we created Protocols IO, I think many of you, if you're academic and have done research and have read research papers, will be familiar with this issue where the research papers tend to describe the results of experiments very well. But when you start to look at the method sections, there is a lot of circular, as you can see in here, method was performed as described in another paper, pointing from one place to another. Here's a biologist who says, I'm currently reading through so many papers to better understand methods, blah, 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 as described elsewhere. Tell me the secrets without all of this noise. And the person says, I'm appreciating the methods detailed on Protocols IO. Here's a physicist who is reading a paper, and the method section says devices were fabricated as previously described, previously described, and the original reference it points to simply says devices were fabricated with conventional methods. So I have bookmarked a lot of complaints over the years that are similar to this. We would spend a whole hour going through them if I was to read them out loud. But it is a very common problem, and it's actually been sort of quantified. There was, starting in 2016, there was an effort in cancer biology papers. There was an effort by Center for Open Science and the company called Science Exchange to independently replicate results from 50 famous cancer papers. And when the initial results of that work came out, there was a lot of media coverage of it. And one of my favorite science journalists wrote about this work that the hardest part by far was figuring out exactly what the original labs actually did. Science papers come with method sections that theoretically should provide recipes for doing the same experiments, but they're often incomplete, missing out important steps, details, or ingredients. In some cases, the recipes are not described at all. Researchers simply cite an earlier study that used a similar technique. So this points to the complaints we saw on the previous slide. And the Center for Open Science on their page about this initiative says that in those 53 cancer papers, out of 193 experiments that they wanted to independently replicate, 0% of protocols were completely described in the paper itself, and it was important to contact the authors, the research labs, and ask them how exactly was the work done. So this was back in 2017. Here we can see this is 2025. We can see that we still have a lot of challenges in terms of reading the papers and how we communicate the science, what's included in them, and there are calls for reform and improving the reproducibility of what we publish. So this is at the heart of what we're trying to improve with Protocols IO. But what I wanted to point out is that what's interesting about this initiative, the Cancer Biology Initiative, is that when Center for Open Science scientists contact the authors that published these 53 papers, when they reached out to the professors and asked for the original protocols, even when the professors wanted to help, frequently they couldn't figure out which protocol version, where it was in the lab notebook of a person who already left, which version was used for the paper. So the problems of replicating and building on previously done work, it's not just the way we publish. What their initiative showed is that the problems start much earlier inside our research teams in terms of how we do the work, how we keep track of it, and that it's often chaotic. And even though we have lab notebooks where we document exactly what we've done, it's often hard to figure out which version of the protocol was used to generate the specific results, and what you should be sharing with others. And more recently, there is a movement to improve publishing, so that the paper is sort of the central piece that's made up, what we call modular publishing, that's made up of pointers to the data, to the code, to the protocols and methods that were done so that a paper becomes a sort of puzzle that fits in from all the different pieces. And on the right-hand side, you can see where we're moving. This is an article from Nature Communications where you can see the protocols are in Protocols IO. The data is in UniPro database, um, and Protein Data Bank, and the plasmids that were developed for the work are at the Adgene repository available for anyone else. So there is a big push by publishers to move in that direction. And what it also helps with is giving people credit for the work that they've done. So we have a whole separate webinar that we run that is about credit and recognition for method development. But if you go to the original Nature Communications paper, um, here is an author who is somewhere in the middle. But when you come to Protocols IO, it's clear that this is the person that developed the method and did most of the experiment so you can get recognized for your contribution in more ways than just on the paper itself. And uh, every single public protocol on Protocols.io, you'll see has a metrics tab where we try to highlight everything we know about how people are using and reusing the protocol, if it's been cited in any other papers. So you'll see the metrics tab on every protocol on Protocols.io. All right. And now I would like to actually switch out of the slides to do a quick demo of Protocols.io itself and the key features. So um, I'm going to show one of the public protocols, but every protocol that you create on Protocols.io starts out privately. You decide who to share it with, if and when to share, what permissions to give to people. So I'm giving an example of a public protocol, but anything you create on Protocols.io is private, either completely private to you or just private in the workspace to your colleagues and collaborators or your research team, depending on where you've created it. I'll show a little bit about that in just a moment. But you decide if you want to make it public and who has access to it. Okay, so when we go to a specific protocol, what you will notice is that they are not PDFs that are like published articles, just fixed in time. The protocols are dynamic and interactive. We have more and more protocols that have videos in them. People are adding tables with formulas. We have a lot of components and structure that helps with reading and understanding the protocols. It's broken down into the step-by-step instructions, the guidelines section with context around the protocol, the materials that are needed and the equipment that was used to do the work, and then the metrics tab that I talked about. The steps themselves are organized into sections. You can quickly navigate to the appropriate part of the protocol if you're doing the work. And in terms of them being interactive, a really important feature that I think is one of the essential and most important things about Protocols.io is that you can, on any step, click this plus button and ask a question or make a comment that will go to the author of the protocol and everybody who's using it. So if you're asking it on a private protocol, it's for collaboration inside the team. So you can ask on a private protocol, or if you're looking at a public protocol, you can ask the question. You always have the option of asking the question privately, or you can ask it publicly, as you can see on step 2 here, where there are comments and questions. So someone is reading the protocol and is asking a detailed question about it. That goes to the author and everyone who uses the protocol. And then you can see a sort of public discussion that's happening right on step 2 of this protocol. And if you go to the top, you can open all of the different comments and you can see that there's protocol level questions just about the protocol. There's step level questions. You can ask a new one. And this, as I said, allows to build a sort of FAQ right on the protocol itself. So instead of a question by email to the author that's hidden from everyone, and lots of people are asking the same question, and you're answering the same question over and over, when you answer in Protocols.io, you can point to this, you can point people to the discussion, and ideally, over time, people stop asking the question because they see the answer right on the protocol. The other important part of the protocol viewer is that you will notice there is versioning. So when we go back to my personal story of spending that year and a half making a change, correcting a protocol, and not being able to let people know about the improvement. On Protocols.io, it's really easy. So even if you've published a protocol, you have a DOI, a document identifier. This is what you use to cite. So if you go to metadata, you'll see the citation for the protocol. There is a DOI link. So if you've shared a protocol, it's fixed. If it's public, it cannot be changed. But as conversations happen on these steps, if you want to improve or if you have a correction or optimization for your protocol, with a click of a button, you can create a new version and share those changes. And there is a compare functionality. So to the right of the title, if I click the three dots, there is a compare functionality that actually lets you see which steps between version 1 and version 2 are the same and which steps are different, highlighting the steps that have changed. So you can see from step 3 to step 8, there are no changes, and then in step 9, there's something different between version 1 and version 2. You can create as many versions as needed. You can keep your protocol current. You can come back to it 2 years after you've shared it and say, actually, we now have new enzymes, new kit, new equipment, or we've optimized how we're doing this, and there is a better way to perform the technique. The previous version doesn't disappear. You can switch to it. But anyone who comes to Protocols.io, if there is a link to the earlier version, when they get here, they get a pop-up that tells them that a newer version is available. So they can view the older one or navigate to the most recent, could be version number 10. Mhm. The other important thing that you'll notice is that every protocol, whether it's public or private, has a copy fork button. So this allows every protocol to sort of be a template. Let's imagine you work with heart tissue and I work with brain tissue. There might be slight differences in how we process our samples for analysis. It's not that one protocol is right and the other protocol is wrong. It's just that they're two very similar approaches with slight variation between them. So that's what the copy fork button is. And every protocol that you see on Protocols.io, out of those 24,000 public ones, you can take it if it looks good, if it's similar to what you need, you can take it, you can click fork, and you will get your own private copy of it. Then you have an edit button and you can make changes because you're working with brain rather than lung tissue, depending on the equipment that you use. So that protocol becomes editable. You don't have to scribble notes and print out the PDF. You can just make a copy of it, right? And then the last thing that I wanted to show on the protocol view level, the last thing that I wanted to show there is the run button. So for hands-on wet lab protocols, and we do have a lot of computational bioinformatics workflows that are not hands-on, but the majority of protocols, the public protocols that are on Protocols.io, are wet lab and field methods. And for those, this run button is very useful. So this allows you, as you're doing the work, to have a sort of checklist app on your phone, on an iPad or a Samsung tablet. You can run through the protocol. You can say, you can see where you are, what you're doing next. You can start a timer in the fume hood as you're doing the work. And importantly, if you change anything as you are running through the protocol, as you're doing the experiment, you can click the edit button and you can say, actually, today, instead of room temperature, I did it at 25 degrees. And when you do that, you have a record of exactly what you did on that particular day, right? And when you're done, you finish, and it saves in your file manager on protocols.io in your workspace in the folder that you've selected. It saves a record of how you did it. So we're not changing the protocol itself, we are changing how we ran it, the run record that is then stored in the folder that you've selected. So the protocol remains, but you have a record of how you ran it on that particular day, right? And now what I would like to show is what happens when you create an account on protocols.io, where you typically are, because most of the time you will be in what we call the file manager inside your workspace. And when you sign in, it looks like, as I said, your folders, your file manager. So here I am in a demo workspace. There are folders, subfolders. You can organize it as you wish, and make sure that the protocols are easy to find. You can organize it by project. So it could be that kidney, lung, tissue could be the folders, or you can have for your team, the organization be protocols that are in development, protocols that work, and protocols that are old. So it's up to you how you organize it. You can have folders and subfolders inside. And these spaces, you can have a workspace just for yourself where you keep the protocols for yourself. But one of the beautiful things about protocols.io is that you can collaborate with others. So um, you can create a workspace and you can invite people easily to join it, and you can have a whole research team or a whole company in the workspace working together, sharing protocols. If someone graduates and leaves the university or postdoc moves on from your team, the protocols remain in the workspace. If someone new joins, you easily add them into the workspace, and they now have access to all of the research methods that are organized from your team in one place. Everyone is using the most recent version of the protocol, and it is a way to make sure that everyone is synchronized and is on the same page instead of loose pieces of paper floating around our bench tops. Mhm. And before we finish up and go to the Q&A, I also wanted to spend just a couple of minutes talking about creating a new protocol. So when you're in the workspace, you choose the folder where you want to create it, and you'll use the new button. And I wanted to mention that in every workspace, you can create in whatever folders and subfolders, you can create protocols in those folders. But every workspace, you also have a folder with your name. The folder with your name is kind of like a private space. This is your private folder. If you create a protocol not in the main workspace, not in the uh, top level organization, but inside your folder, those protocols, even in a collaborative workspace with 30 different people, if you create a protocol in your folder, it's only visible to you. So that's your protocol, it's visible to you, and you can drag and drop it. So you can select the protocol and you can move it to the folder shared with everybody. But if it's in your space, it's only visible to you. And if I switch to the main workspace right there, these are protocols in these folders that are visible to everyone. And now going to the new creation, it's self-explanatory. If you're starting from scratch and you have a new protocol, that is really easy. right? You go into the create new protocol, you give it a title, you will end up, you decide where you're putting it, and you will end up in the editor. So here you can create step-by-step protocols, uh, create sections, you can put in the descriptions, guidelines, warnings, before start instructions. So this is all pretty self-explanatory. On the right-hand side, you will see a history of all the changes. The protocols are have collaborative editing, so you can have co-authors, you can have kind of like in Google Docs, you can have multiple people working in the editor at the same time, and we keep a history of who made which changes, what the changes are, and when they were made on the protocol, right? So most of it is self-explanatory. You will also see on the right-hand side that there are components, and you can add into steps different components. Again, most of it makes sense. You choose whether you are working with a biological protocol, chemistry, or computational, and based on that, you'll have different components highlighted that help to add structure and help to make your protocols more accessible and easier to understand for yourself and others. But if we go back to the file manager, before I stop for Q&A, an important point that I wanted to make is that when you click New, you will also notice that there is an option to import a protocol. And what this means, what this allows you to do is add a PDF, Word, Excel, spreadsheet, and using OpenAI, we will automatically convert it into protocols.io. So instead of going into the editor for an existing protocol and copying and pasting steps one by one, um, you can just drop the file, one or more files, into the importer, the we call it Protocolify, and within a couple of minutes, it will create, based on the PDF, it will create a protocol. It's not perfect. It works well 9 out of 10 times, but then you can go into the editor, check it, make any corrections, but it saves a lot of time in terms of getting protocols into protocols.io. So you'll also notice that I can drag and drop from my computer. I can put drag and drop different files, or I can click new, upload a file. If you have existing PDF, Word, or any other format documents, protocols, if you want to quickly get them into the workspace for yourself or your co-authors and colleagues, you can do that easily. And so it's not just the protocols that you create, but you can also put in zip files, PowerPoint, etc. And once you've put in a PDF, you will notice on the right-hand side, there is the button to turn it into a protocol using the Protocolify module. All right. So with that, we are going to exit out. There's of course a lot more really good functionality that we've added to Protocols.io for administration, for sharing, for collaborating, for working in the editor. There's a lot of functionality, a lot of really good features on Protocols.io, but we'll leave that for the advanced webinar. And I will finish up with just a few slides before we go into the Q&A. I did want to say that even though most of the protocols in Protocols.io are biomedical, we do have a lot of different disciplines, physicists, psychologists, archaeologists that use Protocols.io. We have a lot of people that use Protocols.io for how-to instructions on building instruments or configuring a microscope, setting it up, or creating devices. So These are some examples. Again, we will share the slides. You'll have all of these links, but you can see that we welcome research methods from all different fields. It's not just for biologists, right? And there are more and more publishers that recommend and encourage use of Protocols IO in the author guidelines. I think it's now not just 500, but over 1,000 different journals. All of Wiley journals have Protocols IO and author guidelines, all of the journals from Public Library of Science and the other ones here. So there's more and more journals, more and more funders that recommend Protocols IO. And before we finish up, just wanted to remind you that we have uh, the different webinars, like I said in the beginning. We have the one-on-one demos that you can request at this link. We have very short, this one is a long 45-minute webinar, but we also have tutorials, which are videos of 1 to 2 minutes that show you how to do something very specific, how to publish on Protocols IO, how to manage, add, and remove uh, team members from your workspace. So we have a lot of tutorials, and we also have a really detailed and helpful help center where you can find a lot of information if you don't want to uh, reach out to us. And of course, info at Protocols IO, there's several people across different time zones that work on Protocols IO. And if you send us a question, we will happily and quickly reply back. All right. I will stop here for the Q&A, but also wanted to ask you if you're on this webinar or listening to the recording, if you can take a moment, if you can take a moment and go to this link to give feedback, that helps us to improve the webinars, the content, what we focus on, what we present. So we'd love to hear from you if you can take 30 seconds to answer a few questions about the webinar, and if there was something that you wanted to hear on it that we didn't get to, that helps us to optimize them. All right. With that, I will stop and go to the Q&A. So the first question from a professor is, I'm just going to read it out loud. Would I be able to find in Protocols IO where the material or instrument is available locally in my area with cost and contact personnel and alternatives to that particular machine or technique? So that is a great question. Thank you for asking it. It depends on the protocol. It depends on the protocol. Not everyone shares very detailed information, but what I did want to say is always make sure you search, you do a good search on Protocols IO, and uh, I wanted to highlight that when you do a search, by default it searches protocols. But you can see that you can also search workspaces, and these are public communities. One of the communities that we have is called low cost, high quality methods. Right? So this is a workspace, a public community. They have 30 members, 48 protocols that's specifically dedicated to sharing for resource restricted regions, um, sharing low cost, high quality methods. So um, look for protocols here, but also if you don't see them, join this workspace. You'll see a button on the right that says join. Go to discussions and ask, does anyone have XYZ, that will go to everyone in the community. So check out communities and workspaces. It might not be the specifically the low cost one, but if you're working on wastewater surveillance, take a look at the workspaces. Let's sort by members. Um, here is a wastewater epidemiology workspace. Here is a workspace on poliovirus. So if you work on that, join the workspace and then ask the members of the workspace about uh, different versions of the protocol that might work. Can I use less of an antibody? Can I use a different make your own buffer to do this instead of ordering an expensive one from a supplier? And finally, importantly, don't forget that every protocol lets you comment on it. So um, even if you're not part in the workspace, if you find a protocol that is what you need, but you're wondering if you can do it cheaper, go to the protocol, click on the steps, and ask a question, right? Go to that plus, ask a question, and don't be shy because that will go to the author and everyone who is using that protocol, and they might be able to share ideas for how to do it on a lower budget. But fantastic question. Thank you. And let's see, any other questions in the chat or in the Q&A? In case you're shy, we're going to stop the recording here.